Welcome to FAU's Virtual Research Cafe, where two faculty members from different areas of research talk to us about their work.
I actually began interested in maternal substance use and really healing women in recovery, working as a perinatal and oncology clinical social worker in a birthing hospital in Providence, Rhode Island. And several years ago, before I finished my Ph.D., went to a conference in Nashville, Tennessee, because at the time, in about 2014, it was actually considered a criminal charge if a woman who was pregnant was using substances during the pregnancy. They would actually be arrested and placed in jail. So there was a national conference in Nashville, Tennessee, and I brought my daughter with me, who is now a master's student, a graduate student in the College of Social Work and Criminal Justice, receiving her master's in criminal justice. And it was a conference really to share with lobbyists and physicians and how punitive policies can really negatively impact women in treatment engagement and also disrupting families. And that was kind of where it began, of shifting from working and working with women specifically in trauma, but then also working with women that have experienced substance use. So my focus is on pregnancy and early parenting women, because it's this, we know and scientifically that it's this moment where people are so motivated for change, and not just desisting from substances, it's everything, whether it's a healthier diet or exercise, and really starting to think about different things in their life. And it's this opportune time to really strategically have clinical interventions at this time to help women. So the first study I really wanted to share with you is I wanted to explore a qualitative study of women that were pregnant and had opioid use. What was their decision-making process, and not only in their prenatal care, but also in their treatment of opioid use disorder. And what I discovered qualitatively, that there was this, amazing amount of not only internal stigma, the shame, but also external stigma, feeling, receiving a lot of bias and judgment from healthcare providers. And in the midst of that, trying to enter recovery, while being pregnant, and thinking about, okay, what do I do? I don't want to use anymore. Do I start methadone? Do I start buprenorphine? I want to taper. I don't want to be on any medication at all. Simultaneously, they have this parallel fear of neonatal abstinence syndrome, which is a syndrome that can occur in utero if a fetus has been exposed to opioids, and they have some irritability, and it's a treatable syndrome after birth. And so they had that fear, and then alongside that, the fear of their infant being taken away by Child Protective Services because of the state laws. But what was amazing, that there were two outliers in this small qualitative study of 20 women, that 2 of the women received support from a healthcare provider, and it really was transformational in entering treatment recovery. So then I said, okay, to really want to explore, what is it like when you provide support for autonomy, support that you believe that somebody can have positive outcomes, and can that actually negate the internal and external stigma that is so powerful, and we know is a barrier to treatment engagement. So just really briefly, shared decision-making really just assists individuals through complex decision-making. So intuitively, many of you probably already do this when you're seeking, you have a healthcare decision, you're involved, your voice is heard, the options are told, risk-benefit type of discussion. But there's many times where, especially with substance use, women are really disempowered and not included in the decision-making. But we know that it is an evidence-based practice for really assisting and having positive outcomes for high-risk decisions. So Dr. Friedman and I conducted a US survey. It was a representative sample in the United States. We have about 500 participants of OBGYNs, and we really wanted to just look at how many OBGYNs were doing the recommended standard of care for opioid use disorder, first of all, which is actually a medication-assisted treatment is recommended for women that are pregnant and have an opioid use disorder, either methadone or buprenorphine. And there's many reasons for that. We don't have time for that, but that they're in care, they're receiving care, and it limits some of these other, high-risk behaviors and also traumatic events when they know that they're being seen and cared for throughout the pregnancy. It's much safer. We also wanted to see how many physicians actually utilize shared decision-making. This is just a really quick representation of the United States. You can see there was predominantly on the East Coast that were, answered to our national survey that was sent out, and California. But up to 77% actually had seen a pregnant woman with opioid use disorder in the last year, and only 14% of the sample actually was utilizing the standard of care, which is the medication-assisted treatment. And of that 14, only 47 that did have a waiver, so did take the short course to be able to have a buprenorphine waiver, were actually prescribing to their patients. So that was one issue. The other was the shared decision making. That was really promising because 61% were using shared decision making, and they were actually including the women in the care of what are the current state laws, to be informed, discussing with them options, and really having some training also in substance use disorder. The final study that I want to share with you is a photo voice study that Dr. Colvin and I completed last year, and we actually are submitting a manuscript this week, so that's really exciting. But photo voice uses documentary photography and feminist theory and empowerment theory to really promote social change. And what we wanted to explore was how do pregnant and early parenting women experience motherhood in the midst of recovery and recurrence of use. How does infant separation impact them, and how do they navigate a very complex, really, especially in the early postpartum period, social service case plans when their infant is removed. So you can imagine it was really changing the recruitment, but it was really transformational, not only for Dr. Colvin and I, but also for the participants. And we wanted to also conduct the ACE course, which is Adverse Childhood Experiences, with our sample, and look at how many of these women actually experienced childhood trauma. And it's a health concern in adulthood. If you have 3 or more of ACE scores, it's 10 is the highest. In our sample, the average was 5. Almost half had a score of 6 or more, and 2 of the moms actually had 9 out of 10 of these adverse childhood experiences that run from a parental loss, parental incarceration, parental substance use, emotional, verbal, sexual abuse. So this is really significant, and that's where the trauma piece comes in, of how we know from the literature and from the research that I've conducted, the high comorbidity of trauma, post-traumatic stress, anxiety and depression, and substance use, particularly with women. I just wanted to show you one example from the photo voice study. And this participant actually was an artist, and she drew this picture. The hands are a depiction of actually her hands ripping her heart. She felt that this was her fault, and this separation with her, the depiction of her son. So she took a picture of both of these artistic depictions, and she said that she would sing. She had been incarcerated for a short time in the early pregnancy, and she would sing this country song, Godspeed Little Man, and he would actually fall asleep on her mother's lap, and how difficult that was for her. And this drawing just really, you can identify and understand how painful this was. And this woman had not only past trauma, but present trauma with intimate partner violence. She's actually doing very well now. She's a peer support specialist and is helping hundreds of women and families in recovery. She's in long-term recovery and has since been reunited with her son. So it's a positive ending for her, but it's the takeaway really for me in the research in this journey is developing strategic interventions for women that have experienced trauma and substance use. And again, Dr. Freeman and I are now waiting to hear from NIH, because we're working on a specialized peer support specialist intervention that really supports women and helps them during this difficult time and helps them engage in services and really heal from the trauma. So thank you very much.
Thanks so much, Heather. I didn't mention it in the beginning, but if you have any questions for our panelists, please type them on the Q&A button. And we will go through the questions after both speakers have presented. And I think we have fixed our technical issues. Yeah. You can hear me now? All right.
Yes,
all right.
I'm sorry. I don't know what happened, but I left the web, decided to get in, and now it's working.
Good. So our next speaker is Dr. Lucia Carvelli, who will be talking about a somewhat related topic, but more from a very different angle, which is drug effects across generations. Lucia?
Thank you, Karen. So before going into the science, I was asked to give you a little bit of background, to talk a little bit about myself. So I'm going to share the screen now. And I will tell you that I was born in Italy, in a city called Crotone, or Croton, here in the south of Italy. Then my family moved to Milan, where I did my study, got my Ph.D. in molecular pharmacology. And 2 months after my defense, I jumped in a plane, crossed the Atlantic, and landed in New York, JFK, the most chaotic airport in the United States. But just to change a plane, and the final destination was San Antonio, Texas. I stayed there for 2 years. I worked at the University of San Antonio. And then I moved to Nashville, Tennessee, work at Vanderbilt University, where I met the well-known Dr. Randy Blakely, the director of the Brain Institute here at FAU. There I was promoted as assistant professor. Then I moved up to North Dakota, University, UND, University of North Dakota. I spent there 7 years. After 7 years, I was a little bit tired of blizzards and average temperature of -20, -30 degrees in the winter. So I decided it was time for me to go back to the south. And 3 years ago, I moved in Jupiter, where I work now at the Brain Institute with a joint appointment and the Wilkes Honors College. And so that's basically my life in the last 20 years that I told you in 2 minutes. That's not bad. Okay, now back to science. So in my lab, we are interested in the mechanism of action of drug of abuse. And so you see here in this picture here of the brain, the drugs, they will, once they reach the brain, they will act in specific area of the brain. And those areas form what it's known as the reward system. And this part of the brain is highly enriched with dopaminergic neurons. And so neurons, there are a lot of neurons in the brain. In the human brains, they counted more than 100 billions of neurons. So it's really complicated to study. But we really would like to know how neurons communicate each other's, because that's where some of the drug directly act. For example, here I have depicted is a cartoon of this type of communication between two terminals of two different neurons. For example, if you imagine that this is the terminal of the dopaminergic neurons that will make a connection, an interface with another neurons, there are in this space here that we call the synapses, there are specific proteins that are really important for the correct communication between those two terminals. Some of those proteins, for example, are tyrosine hydroxylase, which is an enzyme which is required for the synthesis of dopamine. And dopamine, it's a molecule that is released by this terminal here, by these neurons. And once it goes in the synaptic cleft, it will go and bind to other proteins located on the terminal of a different neurons. And once those proteins that are called dopaminergic receptors are activated, then they initiated a cascade of reaction that has a goal, as a final goal, the coding, the transmission of the signal mediated by dopamine. The other molecules of dopamine left in this space will be reaccumulate inside the terminals, the neurons, by another protein called the dopamine transporter. Now, it happened that this protein here, it's also a target of drugs such as amphetamine or cocaine. So this protein, the dopamine transporter, or simply that, cannot distinguish between dopamine and amphetamine. It will take both molecules inside the neurons. But once amphetamine goes inside, then again, it starts a different reaction. And we, in my lab, we study how amphetamine changes the activity of these terminals, and ultimately the communication, the interface between those two neurons here. So it will be easier as I mentioned before, the brain is made up of hundreds of neurons and trillions of those synapses that I show. So we really have to simplify the system. And one of the models that we use in my lab is culture of neurons, neuronal culture in petri dishes, like you see in these images here. And also we have a model where we can label the neurons that we are interested in. Our case, the dopaminergic neurons, as you can see here in those pictures, they are tagged with a protein called GFP, or green fluorescence protein. Then we can have both these label of neurons and the culture of neurons can be achieved by using a model system that we use in the lab called C. elegans. C. elegans, there was a nice video here that unfortunately I cannot, it doesn't show now in this format. But C. elegans is a nematode, which is an elegant way to call the worm. And this worm, it's very tiny, very small. When it's an adult, it's only 1 millimeter. So you need a stereoscope to be able to see the worms. And those animals, besides containing a small number of neurons, only 302, and only 8 of those 302 are dopaminergic neurons, they also are capable to respond to the effect of amphetamine by exhibiting a specific behavior. And so we, years ago, characterized this behavior, this amphetamine-induced behavior in C. elegans, and we named this behavior SWIM, or swimming-induced paralysis. So basically, when you put the worms in water, they swim very vigorously. But if in water now you dissolve different concentration of amphetamine, as shown in this graph here, you can see that over time, at different dose of amphetamine, the number of animals that stop moving, stop swimming, or exhibit this paralysis, as we called, increase. We characterize also this behavior, and we found that there are those same proteins that are required and they are important for the communication in the synapses are also required for amphetamine to induce this behavior, sweep. In fact, in animals that do not express the tyrosine hydroxylase, which is again the enzyme important for synthesize dopamine, or the dopamine transporter, or if they don't express the dopaminergic receptors, we lose that phenotype. So now we have a small, very simple model where we have a phenotype induced by amphetamine. So now we can do a lot of type of experiment. And one of the questions that we asked was if amphetamine can give long-term effect. Why we ask this question? Because amphetamine, as you know, and sorry, I forgot to point it out that when we say amphetamine, I include also methamphetamine. It's part of the big family of these type of drugs. So they can be used as a recreational drug, as they can be addictive, or they also use therapeutically. So I'm pretty sure that you are familiar with some drugs named, for example, Adderall, Ritalin. Those are Adderall, it's nothing else than amphetamine. And it's used to treat ADHD, and it's very effective. So kids that suffer of attention deficit hyperactivity disorder, they respond very well with the treatment of Adderall. But the treatment is a chronic. Those kids are treated for years. And so we were wondering whether this chronic exposure to amphetamine would have some repercussion in that synapses, in that communication between the two terminals, especially in kids where the neurosystem is still under development. So we performed this experiment to see if indeed the chronic exposure with amphetamine would give some alteration, some behavioral effect later in life. We collect embryo from C. elegans and we expose for 15 hours either to control solution or amphetamine. After those 15 hours, the drug was completely washed out. And then we put the animals in agar plates where they grow, they feed, and 3 days later, we collect the young adult and we test them for sweep. And so what we saw was really interesting because we saw that animals that were exposed during embryogenesis with amphetamine, and then as adult exposed to control solution, there was no difference with respect to animals that were exposed with control solution to embryogenesis, during embryogenesis, and then to control solution as adult. So the green circle, compare the green circles with the black circle here in the bottom. So these animals, even if they were exposed with amphetamine during embryogenesis, they don't show any difference with respect to the ones that were exposed to control solution. They are fine, physically they are not impaired, damaged. But when we observe the animals that were exposed to amphetamine during embryogenesis, and then again as adults, they are tested into the solution with amphetamine, we saw that the number of the animals that exhibit sweep or they paralyze is higher compared to the animals that didn't receive amphetamine during embryogenesis. So that was really interesting because it showed that amphetamine does something during embryogenesis, which is remembered later in life in these animals and make those animals more receptive to amphetamine, or if you want, more vulnerable to amphetamine. But we went even further. We test also the next generation. So animals that originate from the F0 that were treated with amphetamine, those animals, they never saw amphetamine. they see for the first time amphetamine when we perform this experiment here. And as you can see, also in this case, we see the same trend. Again, animals that originate from parental line that saw amphetamine during embryogenesis are more vulnerable to amphetamine. This was very excited for two main reasons. First of all, it tells us that we have to be careful how we treat our body, because we are responsible not only of our health, but also to the health of our kids and maybe also our grandkids. The other reason why it's really an important data is that for the first time, it recapitulated some epidemiological data that were collected many years ago. So there were some studies where they observed adopted kids that whose parental biological parents, had history of addictions, but they were adopted in healthy families, so they grow in a healthy environment. Even though they observed that those kids had higher chance to become addicted themselves as they became adult, suggesting that addiction has an heritable component. However, this heritable component did not have the classic Mendelian distribution. It could be heredited, but it was not like the hereditance that you see between genes. So now we have the ability to investigate this mechanism in the lab using the simple system as a C. elegans. So one of the first questions that we can ask, for example, is are those proteins that I show you initially that are required for amphetamine to induce the behavioral effect sweep, it's the function of those proteins altered after those 15 hours of treatment during embryogenesis. So that's what we did in this experiment here, where we went and checked the function of the dopamine transporter. Remember the protein that is important to reuptake dopamine inside the neurons. And we found that indeed in culture, dopaminergic culture that originate from progeny that of parental line that were exposed to amphetamine, they have less capability to uptake dopamine inside the neurons with respect to the control, the blue bar, the animals instead that did not receive amphetamine during embryogenesis. So yes, the function is altered, but the question now is how amphetamine that it's given during embryogenesis, and then later in life, the animals are fine. They don't show any impairment. They can swim fine, but they kind of been primed. You see this purple dot here? They've been primed. And when amphetamine come in place, they succumb. They cannot swim and they paralyze. So how this is possible? So one of the mechanisms through which amphetamine maybe can act is through the altering epigenetics mechanism by controlling gene expression. So I'm sure that many of you knows that all the genetic information, all the information to form an individual and human being as well as any animals is stored in the nucleus in this long double helix molecule of DNA. And beside the DNA, there are also a special group of proteins called histones. And so you can see that the DNA is calling around, it's wrapped around those histones. And the binding between the DNA and those histones can be tight or can be a little bit loose. So when it's tight, the information that is in that specific sequence of DNA is not going to be translated in a protein, is not being expressed. When it's instead a little bit loose, that the DNA can be accessible to other proteins, such as transcription factors, that now can come and transcribe the information from the DNA to the mRNA, and then ultimately to a proteins. And so we went and checked if amphetamine can alterate that binding between the DNA and the histones. How we can do that? What regulated this being tight or a little bit loose between those two components, DNA and the histone, are specific chemical groups like methylation group, methyl group, or acetyl group that go in and bind to specific residue of this protein. So we can measure how these change of groups happen by performing a particular type of experiment called CHIPS, or chromatin immunoprecipitation. And so as you can see here, those are the results of this type of experiment. The sample that were collected from animals that were exposed to amphetamine during embryogenesis, the red bar, are statistically much lower, reduced compared to the sample that were collected from control. And we are detecting specific markers of those histones, like in this case, the H3K4, H3K36 trimethylated. Instead, there was another markers, the H3K9 dimethylated, where the opposite was true. Now these markers were increased following the amphetamine treatment. And the same trend was observed also in sample that we collect from the F1 generation. So the increase of these two specific markers and the decrease of these other markers is associated with gene silencing, meaning that when those markers are present in the specific area of the DNA, in our case, we tested the area that expressed the gene for the dopamine transporter, that gene, it's not likely to be expressed. The expression of that gene, the transformation of that gene, the translation of that gene in a protein is not going to happen. It's less likely that happen. And so meaning that you will have at the end less protein. And this explains that functional data that we collected that I showed you before, where we found that the animals that receive amphetamine during embryogenesis have reduced capability to uptake, to accumulate the dopamine into the neurons. And that's because there is less protein, less dopamine transport. So what's next? So next now there is a lot of other questions, because the most complex question is how those changes can be transmitted from one generation to another one. And what I told you so far is that the effect of amphetamine is primarily in neurons, but how the information that amphetamine gives in those neurons can find its way in sperms or oocytes. Those are what we needed to create a new individual in animals. Even C. elegans, that is an hermaphrodite, uses sperm and oocyte. She produces both. And so does amphetamine diffuse and can act inside the sperm or oocyte and change the epigenetic landscape? And if this is true, what is the role of the dopaminergic neurons? Do they have rules at all in the transgenerational effect? And so that's what we are trying to answer. And now in the lab, we are performing an experiment to try to figure out how this transgenerational effect of the drug happen. And I will stop here to take questions.
Great, thank you so much, Lucia. Just as a reminder, if you have any questions, please go on the bottom of your screen, go to the Q&A button there and type your question. And it can be for either Heather or for Lucia. Okay, Heather, can you turn on your video again? And because the first question is actually for you. So how do you balance keeping maternal connections intact and protecting the child's safety?
Yes, that's such an excellent question. And obviously safety is paramount. And, you know, we've just seen an increase nationally in the data of more infants, up to 30% across the country, being placed in foster care due to substance use. So it's a major concern. However, we know scientifically that a healthy attachment between, you know, from the attachment theory between a parent and infant is so crucial. Usually it's maternal, it can be also paternal. So when you have that separation and that disruption, it's very difficult. So the data from SAMHSA, National Drug Use and Health Survey, there's really only about 19% of women are actually able to find treatment during pregnancy and early parenting. So there's such a lack of treatment for this population, and even less to keep the mother and infant as a dyad. So when you're supporting that relationship and you're putting in resources, housing and education and employment, all the important socioeconomic factors that are also so important with somebody's recovery, we call it recovery capital, can have really transformational outcomes with where you're really disrupting this intergenerational trauma. Because if you remember that there's such a high incidence of these women having traumatic experiences in their past. So how important it is to have these very strategic interventions for the mother-infant dyad and really work on that relationship. So I hope that answers the question. Safety is very important.
Thank you, Heather. Okay, our next question is for Lucia. Is there any challenge to generalizing C. elegans to developing human babies?
Well, yes. And one thing that I forgot to mention, and I'm glad that this question was asked, is that C. elegans is an hermaphrodite, meaning that they produce both sperms and oocytes, so they don't need to mate with a different organism. So the genetic pool, it's always the same. With human beings, that's not the case. We are non-hermaphrodite animals, so we always mate with a different individual. And so the probability that amphetamine can give those effects, it's much less. So if the penetrance of the effect that we see transgeneration in C. elegans, we will expect that in humans will be much reduced. So we'll still expect that also in human beings, there will be some transgenerational effect as the epidemiological studies show, but it will not be so penetrant as in C. elegans, meaning that less progeny will have that tendency of being more vulnerable to amphetamine.
Great, thanks. Another question for Heather. You mentioned that story with the woman who was in jail and was singing to her son, and the question is related to that. And other stories similar to that. How do you as an individual refrain from getting emotionally wrapped up in these stories that can be so heart-wrenching?
Yes, oh, that's such a great question. And, you know, especially as a qualitative researcher, we use reflexivity, and it's constant, you know, and really reflecting on, you know, how are you interpreting the data? What are your implicit biases? How is this impacting you? How might you as the researcher be impacting the participant in the study? But, you know, not that particular person, but another participant that was in that study, she said, the first time that we met in the focus group, you said this was going to be an empowering experience, and I didn't believe you. But after the third focus group, we had 3 focus groups consecutively, she said, that was one of the most empowering experiences I've had in my life. So it's wonderful that the research actually also has this dual purpose, that, you know, that it's promoting hopefully social change, but it's also empowering women's voices. And I find such really actually joy in the work, because I see women starting to love themselves and believe in themselves, and believe that they're able to be a mom, which is most of their goal, is that they want what's best for their child. So it's very, very rewarding. And like Dr. Scarpinato, I'm a runner. So running also helps.
Yes, we see each other on the trail once
At A1A.
Yep. Okay, the next question is for Lucia again. The attendee is curious about the transgenerational effects of dopamine. How might this occur in terms of behavior or physical symptoms? Excuse me.
I don't know if I understood the question. Can you repeat, please?
The transgenerational effects of dopamine, so one generation to the next. Does that have any behavioral or physical symptoms in the next generation?
So the transgenerational effect is from amphetamine, not from dopamine. And so the data that we have so far, as I show in the graph, the animals, they don't show any physical impairment, any physical damage. They are fine, like the others that were not treated with amphetamine, and until we challenge them with amphetamine. And so only when they are under that strenuous condition, so when the worms swim, it's not something that they really like to do. They prefer to stay in a solid agar plate, but we force them to swim. And when they swim, they release dopamine. And now if they have a normal function in dopamine transporter, the dopamine will be reuptake and they can keep swimming. But because they have less expression of the dopamine transporter, then the dopamine cannot be reaccumulated as quick as in the control animals. And the accumulation of extracellular dopamine will make them too paralyzed. And so that's the key of the effect of amphetamine. It doesn't give an effect that you will recognize right away the impairment, the deficit. The deficit show up under stressful condition when you require the effect of the protein, in that case dopamine transporter, to be 100% able to function properly.
Great, thank you, Lucia. Okay, another question for Heather. The attendee recently participated in an interview for a research study on how trauma affects speech and language development. The question here is, have you worked with anyone that looks at child developmental outcomes related to these issues, specifically the child developmental outcomes based on the mother's past trauma or mother's point in her addiction journey?
Yeah, that is a great question. And one of my colleagues at University of Miami, Dr. Emily Banstra, she has been doing that research for decades and actually conducted it with neonates with the crack cocaine epidemic in the 1980s. And the results specifically to the maternal substance use of crack cocaine were, didn't demonstrate significant delays. They were really, within just average, maybe a little bit below average. And what she really learned was how the environmental factors were so critical in the development of the early childhood, with just attention and healthy attachment and reading and all those. So, but the public health data with the adverse childhood experiences is just really important to explore and think about how these experiences impact then later on, subsequently, when they become adults and they become parents. So modeling healthy relational attachment for these new moms is in this critical time from, with 0 to 3-year-olds is critical. But what happens when there's separation, that is actually the trauma versus the substance use, if you will, because they've had this disruption and they don't have the healthy attachment. They've been to different placements unless they are able to have kinship placement. It's very complex. So it's a great question. I think that I've worked a lot with the National Early Childhood group in DC, and they're seeing in all the early Head Starts some like behavioral disruptions. And so they're starting to look into how can we support families where infants have been exposed to opioids. And it's critical. So thank you for that question.
Great. Thank you, Heather. Okay, the next question is for Lucia again. Do you try testing these worms with anything other than amphetamine?
Yes, we did. We did basically the same experiment using cocaine, and the results were slightly different. So we had to use a higher dose of cocaine to get some result, and even in that case, the result, they were not so penetrant and so strong like we did with cocaine. So it can be that the two drugs act differently, but I also have to point it out that it could be simply due to the fact that the affinity of cocaine to the C. elegans dopamine transporter is less. It's not as strong as the affinity of amphetamine to C. elegans dopamine transporter. So maybe it's just a chemical issue.
Great, thank you. So we have one more question. That one is also for Lucia. You mentioned environmental stress playing a role in how amphetamine affects C. elegans. Is it conceivable that certain chemicals block how organisms respond to stress, or do we minimize the environmental stress?
Well, for sure. As I told you a little bit about those epigenetics mechanisms. So as you see, they control gene expression. And everything we do, we engage different type of proteins. So maybe, for example, in this moment, while I am talking, I'm using a different setting of protein that I normally use if I'm just typing the computer. And if I keep talking and talking, maybe those protein at a certain point will run away. So my gene need to be producing new protein. So there we go. There are epigenetic mechanisms that will tell new proteins are needed. And so everything we do, we will require this, will engage this type of mechanism. And of course, stress is one of the most powerful environmental factor that will trigger those type of expression. And so stress has a big component. And so it will reset everything. Is a database, it's normal. It will be completely reverted in terms of stress when stress comes in place.
Great. Thank you so much. Thank you so much, Heather and Lucia, for being our first in our virtual research cafe. This is going to be a monthly event where, again, we are having two speakers from different disciplines, mostly different disciplines, speak on their research, and really with the intent to try and learn what's really going on across campus in terms of research. So thank you very much, Lucia and Heather. It's been my pleasure to have you. And we hope to see every one of you back next month when we're having our next research cafe. Thanks so much. Have a good evening. Bye-bye.
Bye.